We have slides and tech resolved. So this morning, Elli Cryan from here at Davis will be talking about evolution of maize hybrid incompatibility. And as always, please feel free to put questions in the chat, and Elli can get to those at the end of her talk. Elli, I think we're at time and people are showing up, so feel free to take it away.
Okay, can everyone hear me? No.
Yes.
Oh, awesome. Thank you. Yeah, good morning, everyone, or good afternoon, depending on where you are. I, like Jeff mentioned, am a student in the Raspberry Lab, and today I'm going to be talking about a project that I haven't talked about in this meeting before. This is a project that's been in some form or another going on in the Rossi Bar Lab for maybe 5 years now, but I started on this project maybe a year and a half ago. So today I'll be discussing evolution of a maize hybrid incompatibility. So the gametophytic factors are a type of hybrid incompatibility you may have heard of before if you're deep in the maize community. And we'll be talking about them in the Zea maize subspecies, but also in some related species. And before I start, I just want to acknowledge some people who have helped me a lot with this project. So the Rossi Bar Lab, specifically Garnet, who started as an undergrad on this project and is now a staff in the lab, a lab assistant. And then I'm also co-mentored by Dan Klebenstein in the Plant Sciences Department here at UC Davis. So we don't have a nice chalkboard drawing. So the Klebenstein lab gets this giant broccoli instead. Today, I'll be going through a little bit about the background of the Zea maize species. Then I'll be talking about the gametophytic factors in Zea maize, and then going through some results that we've had about how the genes that control these gametophytic factors evolved, and also how the haplotypes or genomic backgrounds on which these genes reside evolved. So you may be familiar, more than most audiences, with Zea maize, which is the species that includes both corn, modern maize as we know it, and teosinte. This is a field we planted for this project.
The brackets back to us by noon tomorrow.
So,
what?
So this is a field we planted out a few years ago. These are some hybrid teosinte maize plants that we collected tissue for. So what we know about maize is that the two teosintes, or the two main teosinte subspecies, Parviglumis and mexicana, can pretty readily hybridize, and they can also pretty readily hybridize with modern maize. If we think about what this might look like, we kind of have this really rough tree that I drew where we've got this fork of 3 different groups that are about equally related to each other, and then an outgroup of Zea diploperennis. Also recently from our lab, we've got this 2 teosintes made modern maize paper showing a little bit more detail on how modern maize is kind of a combination of Parviglumis and mexicana, and that the amount of admixture in this kind of varies across different regions or different traditional varieties, but it seems that there can be up to almost half and half maize and Parviglumis contribution to modern maize, mexicana and Parviglumis contribution. So if all of these different subgroups can readily hybridize, how do these subspecies stay distinct? Like if we can get these fields that are this kind of hybrid teosinte maize, and teosinte and maize grow alongside each other in the origin of domestication, then what's actually keeping these wild subspecies and modern maize separate? So one way that these species have been theorized, or subspecies have been theorized to be distinct from each other, or a mechanism that allows that distinction, are the gametophytic factors, which have been found in Zea maize and a few of the different Zea maize subspecies. So as long ago as 1901, maize breeders were reporting some evidence of some non-Mendelian segregation of certain traits in maize that seem to have to do with some sort of hybrid incompatibility. And so Maize geneticists have been interested in this system for a long time, and the way that this system works, or the reason it gives rise to things that look non-Mendelian, I'll walk through this nice example from Liu et al. 2020 that shows these ears and their phenotypes as an example. So if we cross two wild type individuals that have none of these gametophytic factors or loci active in both the female and the male parents, we get this nice healthy corn where we see full seed set. If we have one of the gametophytic factors active on the female side, so for example, TCB1 or teosinte crossing barrier 1 active in the female parent, and we cross wild type pollen, we'll see that none of these seeds really develop and we get no seed set. So this is an example of one of three identified kind of fully functional or seemingly fully functional gametophytic factors. So the other two are called GA1 and GA2. And so we can see here that when we have GA1 active on the female parent or GA2 active in the female parent, we see at least extreme reduction in seed set. And one thing you can notice in this panel D with the GA2 female versus crossed by the wild type male is that we do have some nice seed set near the tip of this ear. So this is where the silks are shorter. And so if we think about what these factors might look like, well, one of the first questions, just seeing this picture, this evidence is, are these factors all just the same thing as each other? But to get full seed set for any of these female plants, you really need to have the male plant have the compatible pollen. You can get a little bit improvement in seed set, having, for example, a GA2 active female crossed with a GA1 active male, but you won't get that full seed set that you're looking for as a breeder. So here from this paper in Liu, Carmichael, and Evans from 2014, we can see a bit more detail about what's actually going on here to create this barrier. And what we see is if we cross wild type pollen to these females that have different active barriers, either no active barrier or these different three, we can see that the pollen tube has some wonky growth patterns in all of the non-wild type examples. So here, this pollen tube is just really thick, a lot more investment or energy than you would expect to see in a normal pollen tube. In GA1, we see these kind of like crooked bends. And in GA2, we see, it's kind of hard to see, but a bit more dramatic bends, less frequently, but a little bit more kinked here. So thinking about what this does to the potential for a seed on this cob to be fertilized, if you think about the different silk lengths, the silk at the tip of the ear is just a lot shorter, so there's just a better chance that a wonky pollen tube will be able to make it all the way down the silk. So that's the gametophytic factor system, and we're very lucky to have like many, many years, just decades and decades of really great genetic studies on this system, on this set of three loci. But it turns out that they're pretty complicated loci. So we kind of wanted to go in and see if we could pull out the specific genes now that we have better full genomic information and think about how these genes evolved to create this hybrid barrier. So one thing that we had already known is that from a few published reference examples of GA1 TCB, and some information about GA2, we knew that GA1 and TCB1 have very similar gene sequences and that GA2 seems to be the outgroup. If we think about a timeline here of when these might have evolved based on sequence dissimilarity, there's a previously published estimate that GA1 and TCB1 diverged about 170,000 years ago, which is much earlier than maize domestication, which was about 9,000 years ago, which kind of calls into question whether these barriers are actually acting in any sort of way that would create like a species separation between different teosintase and maize. And then we also ran a quick sequence dissimilarity estimate to get an idea of when GA2 might have diverged from GA1 and TCB1. And we think that that happened about 10 million years before present, which is even further outside of this timeline of maize domestication and even outside of teosinte radiation or diversification. So to kind of get a better picture of what's going on in these genes, we wanted to expand our search or kind of broaden our sampling in terms of the species we're looking at to try to capture better what's going on beyond this 10 million year time point before this earliest divergence. So we went through and sampled a bunch more species. Like here's some sorghum. We've got some other types of teosintase, and looking like more widely at the Andropogoniae. Luckily, our lab is also involved in this huge collaborative effort to sequence many different Andropogoniae species in this PAN-AND project. So we looked through a bunch of different Zea genus species, as well as some related species and more of these core PAN-AND genomes. And in terms of just the Zea maize species we looked through the NAM panel, a couple MO17 and W22 assemblies, an A188 assembly, and also PB860, which is a new genome that our lab has assembled. So this is a maize line with active teosinte loci. So the TCB1 and GA2 crossing barriers are fully active with both a male and female factor. And the GA1 is partially active with the male factor, so it can pollinate things that have the GA1 barrier, but it doesn't create the barrier itself. And you'll be able to see why it doesn't have that female gene active a bit later. So this line was bred by Jerry Carmichael and assembled by Arun Seetharam, both who have been obviously instrumental in creating this genome. And I feel like this genome is really nice comparison in our analysis to see an example where all three barriers are active, which is pretty rare to find, or we haven't really seen in any of the other genomes we look at. And this is just a quick dot plot to show you can kind of tell where the different introgression areas might be, where the teosinte kind of got crossed back into this maize line. And for example, this is chromosome 4, the short arm of which we expect to find GA1 on. So this is kind of the TCB1, GA1 area, and you can see that it probably has some teosinte material there, which explains how we see those two barriers active in this maize background. So this is kind of like a giant summary of all of these different species we were sampling. And I have marked some interesting time points in context of the species sampling we're doing, but also just to kind of anchor us to think about when we expect these genes to diverge, if we think that they're actually active in keeping species separate from each other. So about 12 million years ago, we had this split where on one side we have the Tripsicum and Zea genera, and then on the other side we have a lot of other Andropogoniae individuals, including things like sorghum, which is another commercially important crop. And then if we zoom in on the Zea Tripsicum side, we see that Tripsicum and Zea, we think, diverged about 500,000 years ago, based on some previously published literature. So when we compare that to the 170,000 year ago estimate for TCB and GA1, we would expect to only see TCB and GA1 on this Zea genus side. And then when we think about this 10 million year time span for GA1 TCB diverging from GA2, we expect to only see GA1 and TCB here, but see GA2 in probably like all of this area. So we went through, this is again like one slide summarizing a lot of work, but we went through and pulled out the different genes that are representative of each of these gametophytic factors across this entire tree. And we did indeed see that we find evidence of both male and female gene pairs in sorghum and some of these other species that are 12 million years diverged from Tripsicum and Zea. And then in Trypsicum and Zea, we find evidence in Trypsicum of the GA1 locus, as well as in some non-maize Zea species. And then within this Zea maize subspecies set, we find all three barriers in all three subspecies. So this tells us that GA2 is a lot older than we thought it was before. It might have a lot more to do with just like a general type of crossing barrier or reinforcement rather than a maize specific or teosinte specific barrier that keeps these different subspecies somewhat distinct from each other. So that's where the genes are, but when we were looking at the genes, the loci were pretty complicated, which is why these genes haven't really been identified on this like broad scale before, outside of just one reference gene sequence at a time. So to kind of capture what's going on in these loci, to start thinking about what the history of these genes might be, we went through and tried to first think about these larger gene trees, and then next we'll think about what they look like on a haplotype background. So this is a tree of all of the female genes. So this is the part of the factor that's expressed in the silks. So we can see that all of these GA2s kind of group together on this branch far away from the GA1 and TCB1. We see that we have a Trypsicum GA1 that seems to be equally related to the GA1 and TCB and maize. And also unexpectedly, we see these very old inactive branches of GA1. So that was surprising. And we did also kind of think about what this meant for our larger gene tree. So this like rough sketch of what we expect these genes to look like in terms of how related they are to each other is we expect TCB1 and GA1 to group together and then GA2 to be an outgroup. But in this tree, it looks like we actually have this like set of old GA1 genes, these inactive GA1s. They're like a very conserved sequence, even though they're inactive, and they seem to actually diverge from a branch subtending the split between TCB1 and GA1, which is kind of surprising. So then checking out what the male genes look like, we expect to see this pattern mirrored in the male genes if this is like a true pattern we're seeing in terms of how these factors function. And so we also see some very similar patterns here where we see the GA2 very far away from the Trypsicum GA1 and the maize GA1. And when we do look closer at our like GA1 species, we see this branch that's like pretty separate from the rest of the active GA1 that represents these old inactivated GA1s that have, again, a very conserved sequence. So again, we see this change in our understanding of what this gene tree looks like. So the takeaway from our gene evolution side of this is that these genes are much older than we thought they were. So originally, these had been theorized to just be genes contributing to the division between the different subspecies of Zea maize, but we see that these are on a time scale much, much older than the divergence of the different teosintase from each other and maize from those teosintase. And we also see the unexpected, very old GA1 inactivated gene. So to put this in context, we're going to look at the haplotype evolution. So we're going to place these genes onto their respective backgrounds to get a sense of what's going on kind of on the ground in these genomes. So to do this, to kind of think about where these different loci were going to be, we went back to the genetic maps. You can find a lot of these on maize GDB. So we went through to look at where do we see the loci for each of these. So GA1, TCB1, and GA2. And knowing that these genes are very hard to ID or haven't really been annotated in the genome, at least for some of these loci, we wanted to say what is kind of the edge of what we would consider any of these loci. So we went through to see on either side in the genetic map, what is the closest kind of map location or locus where we could find a gene that has been like annotated and well ID'd, and kind of use those as flanking regions to pull out an area that we would consider an area that should include the locus we're looking for. So to do this, I had a ton of help from Garnet, who used to be an undergrad in the lab, but is now staff, and Arun Seetharam, who has been a great collaborator and really helped a ton on going through these genomes and pulling out what could potentially be genes. So Garnet started by kind of using these hits that Arun had found for where these genes could potentially be and organizing them based on their location into kind of an idea of what the haplotype would look like on each background. So for example, if we look at this W22 assembly here at the bottom, we see 2 gene hits here. And then, so these 2 would be, I think, the female gene hits, and this would be the male gene hit. This kind of gives us an idea of which of these haplotypes look similar to each other. So for example, if you look at this W22 haplotype, it looks pretty similar to the 2 right above it. So then to make that a little bit more clear, we kind of looked at the gene trees to kind of label or color different gene hits by which ones were the same or similar to each other, most related to each other, to create these kind of visualizations of what these haplotypes look like as kind of like fingerprinted color-coded haplotypes. So you can see here on the left, the haplotype with the color coding, and then on the right, we have these different gene trees where we've separated out the clades to have the same colors as each other. So one thing we can notice looking at this GA2 haplotype example, in this presentation, I'll be showing GA2 and GA1 because we don't quite have TCB together yet. So for GA2, we can see that this haplotype you find in B73, you also find some pretty similar haplotypes in KY21, Ohio 43, and so on. So you can kind of see how some of these haplotypes kind of group together, where some of these maize lines have like kind of a unified haplotype. And you can also see that there's these like larger kind of chunks where some of them look a little bit more spread out or more like confetti than others. And some of them seem like they don't seem super functional if they only have like 1 hit at a time. So we're going to zoom in and think about what's going on in these different haplotype groups. So this haplotype group with this blue female hit is the group that seems the most likely to be functional. It includes the genomes that we know have some potential functional GA2. And it seems like there's kind of a few variations of what a functional haplotype could look like. So not just one conserved functional haplotype, but a few different options. We've also found that looking in detail at these hits, the potentially functional male hits are methylated and the potentially functional female hits have gene expression just lower than usual. And what I mean by potentially functional is that the genome or the genotype does not display the crossing barrier, like it does not show the phenotype of the crossing barrier, but the gene sequences or the coding sequences are either almost or completely identical to the coding sequences of functional hits. So this group contains a mix of phenotypes, but the genotypes in terms of the gene sequence are very similar to each other. On this group down here, we are looking at this and it looks like there's just these male hits and no female hits. So these like red hits are the male hits, which begs the question, where's the female gene? And we actually found it more than 50 megabases away, which we did not expect. Kind of a hallmark or understood quality of these loci is that the male and female genes are close together and very tightly linked, to the point where people used to think there was no possibility for recombination between the 2 genes, the male and the female. But we find this almost completely unlinked. On the long arm of ZMA's chromosome 5, we find this distal hit. And at least for the genes that we looked at, all of these distal hits seem to be non-functional in the sense that if the gene were to be transcribed and translated based on what our understanding of the coding sequence is, it would be an inactive gene or like a non-functional protein. So this was pretty surprising. We didn't expect to see this. And if there were a functional version of this gene at this distal area, we would expect that to mean that you could potentially see separation of the female barrier and the male kind of ability to overcome the barrier, which could give rise to an individual that would have the female barrier and no way for the male gene to overcome the female barrier, which would create a self-incompatibility. So yeah, this is just going a little bit more in detail in the text on the slide instead of me explaining it with hand motions. So if these two are unlinked, the female function could be easily removed, which would give you an active female gene and an inactive male, giving rise to self-incompatibility. There have been some theories that this system could have an origin as a self-incompatibility system, and GA2 is the oldest locus of these three in the sense that this gene sequence or this syntenic location of the locus seems to be the most conserved and oldest across this like 12 million year spread of species we had sampled. And this distal hit we observed here in both some of these maize lines, but we also saw in some of the other Zea species. So our summary for GA2 is that regulation may be playing a role in repressing functions. So we sometimes see genes that look like they would be active, but they're on backgrounds where they don't create a barrier as a phenotype. And we also have this, what we think is the first observation of a distal female gene. So where the gene is far outside of what we would consider the traditional GA2 locus. Now, taking a look at GA1, which is on the short arm of chromosome 4, we again color-coded these different haplotypes or color-coded the genes on these haplotypes by kind of grouping together these functions. And you'll notice here a lot of the color coding is gray. So everything that's color-coded gray is a gene that has stop codons in the middle of the coding sequence. And so at least in terms of protein function seems inactive. But as you can see from this chart, we have seemingly very strong conservation of this non-functional haplotype where every gene of these three in this haplotype seems like it should be non-functional because of these stop codons in the middle of them. But if they have stop codons, but they're also really conserved, does that mean there's some function we don't understand yet that this haplotype or these genes have? So one thing that could potentially be going on here is as observed in this 2014 paper from Lou Carmichael and Evans, sometimes if you cross this old GA1 genotype to other backgrounds that have an active barrier, you can get over many generations a slow silencing of this barrier. So in this paper, they did this nice experiment where they crossed the active TCB1 to a background that had inactive TCB1, so the wild type, and then this like inactive sugary 1. And they used the phenotype of the sugary 1 across many crosses to a background with the active locus to see whether you continue to have non-Mendelian segregation in the phenotypes of the kernels. So all the way back in 1901, the original way that these factors were noticed was this non-Mendelian segregation in the ears. So by looking at the percentage of the sugary kernels that result from like back crosses to individuals that have the active barrier, you can get a sense of how active the barrier remains over many generations. So the background that they used for the non-functional version of the barrier was W22, which when we looked at our haplotype groups, W22 belongs in this non-functional background. So over like 10 generations, you see complete silencing of TCB1 when you're crossing into a background that has this conserved non-functional W22. So we think that this may somehow be playing a role in slow silencing of other loci when the locus is in the same genome as another active barrier. So yeah, the theme for this project is all of these results are confusing and strange, but kind of exciting. So this definitely is in line with the rest of the types of conclusions we are coming to. Okay, so to zoom out again, thinking about another group of these GA1 haplotypes, this kind of confetti set of haplotypes that have this red version of the female gene are the haplotypes with known functions. So they either are fully functional GA1, where both the male and the female version of the gene seems active, or they're haplotypes that are known to have the male function, so they can overcome the barrier but no female function, so they don't create the barrier. If we look in more detail here, actually the only one of these members of this haplotype group that creates the barrier, so has the active female function in terms of the phenotype, is HP301, which is a popcorn in the NAM panel. This HP301 haplotype, you can see, looks in a way pretty similar to some of these other haplotypes. Like it has a similar set of genes, like where these genes, this purple gene seems present in a lot of these different individuals or genomes, and this red gene seems basically identical in many of these other lines. But when we zoom in to look at what that female gene looks like, seeing that this female gene here looks basically identical in sequence to all these other female genes on these backgrounds that don't have an active female function, it seems like the main difference between the female genes here is that the HP301 female gene has much higher methylation than the female genes, the other female genes in this haplotype set. So we think, although we don't have any further evidence, we just have the kind of observational evidence here of like what these phenotypes are, what the haplotypes look like, and what this methylation pattern looks like. We think that potentially methylation could be driving the presence or absence of the female phenotype here. So our conclusions for GA1, we have this ancient, seemingly non-functional haplotype that is very conserved, so it may have some sort of function in repressing other loci is our best guess for what that function could be. And then methylation may be regulating the female gene function in this GA1 locus. So that's, I think, the overall summary of this project, and some of these like kind of confusing but interesting conclusions we're reaching, seeing these complicated loci and all of the strange things going on in them. So I'm happy to take any questions and hear any feedback you all have about this project or these loci.
Yeah. Let's see. So we have questions in the chat. I think first from Ed about gene expression in the different tissues. Are you sure you have the right translation start site for the non-functional? Or could this be a UORF?
Yeah. That's a great question. So luckily for us, there are some good studies where the tissue types that are being sequenced, or the tissue types that have RNA extraction and then sequencing are getting like more and more detailed all the time. And when you look into the tissues where we see expression, I'm purposely going in and pulling out tissues that are the gametophytic tissues, or as close as we can get to that. So trying to look at like just specifically like silk tissue or pollen tissue or like anther tissue to try and see whether these are active. I think one of the strongest pieces of evidence we have in terms of the idea that these are actually being expressed is that the phenotypes tend to at least generally line up with whether or not there's expression of these genes. Like if you see high expression in that female gene, you tend to see the active female version of the barrier. So although again, these are kind of observational, at least on our side, other labs have studied whether or not the expression actually leads to the phenotype. And we do tend to see that, at least in the genomes we're using that have gene expression data for the gametophytic and silk tissue, that the high expression lines up with the presence of the phenotype.
Yeah, I guess I was kind of wondering if in some species, essentially the GA1 is being used in some other tissue for potentially a different purpose.
Oh, yeah, great question. So I think I also looked to see if we saw activity in other tissues. And yeah, a nice feature of the genome browser on maize GDB is you can look at the expression in a bunch of the different tissues at once. And I didn't do any sort of like in-depth analysis, but I did go through and check like a random subsample of the different lines to see if the gene expression seemed to make sense, and the non-gametophytic or like non-silk tissue seems to have basically zero expression of these genes. So I do think that they're
So across the entire interpogons, they're not. It really is just gametophytic expression.
Yeah, it seems to be, and we are gonna what Garnett's in the process of doing now is pulling out some non-gametophytic factor pectin methylesterases, which is the gene family these genes belong to try to kind of compare what's going on there. And we do kind of expect to see probably some differences given that those are expressed in non.
And then how do you know the translation site's right?
Good question. So for some of these genes, we're also trying to look to see if maybe we've got like the introns wrong or something. Maybe there's like some cryptic splicing going on. But when I was looking through to see if there was any evidence of that, it didn't really seem to make sense. And also the alignments that we have, like the coding sequence alignments, the protein sequences for the published versions that have been functionally validated are extremely similar to each other. So I think like the TCB1 and GA1 male amino acid sequences are only 9 amino acids different from each other. The coding sequences seem to be very well conserved in terms of like the structure. They seem to have like the same introns and like a pretty conserved.
Yeah, I mean, I guess you could have with a U or you could still have a very conserved 5' UTR region, and and I guess unless you had real protein sequence to sit there, or you had riboseq to say that these are it's not being primed there. I guess, yeah. I mean, yeah. I'm not sure how much proteomics backs up any of these studies to show really what's being translated. So I think that would be the other question I would ask is whether because it may be very difficult to tell from bioinformatics between these two.
There's been some pull down studies for like the reference genes for a few of these, and I think that that's like the best proteomics evidence there is. To be honest, it would be nice if there was more evidence on the mechanistic side, because right now part of the model is that both genes are PMEs and that they somehow bind to each other, but there's no evidence anywhere of like two PMEs binding to each other. So seemingly there's like intermediate proteins that bind these things together. I guess the last thing that I would think might be good evidence that we have the coding sequences correct is when you look at the high expression level, it is very high. So you can see that the coding sequences are expressed and the introns aren't, which would make me think it's not a U, or at least for the ones that have good expression data.
Well, I guess I don't know how you tell that from the RNA. I mean, you may have the expression right, but you can't, yeah, I guess I'm not seeing how you're going to figure that out just by looking at the RNA. I mean, you have this. I don't have any doubt that you guys have the splicing correct. It's whether or not there's where translation really starts. I think the trickier one.
Okay. Well, thank you. I'll definitely think about that. I'm sure that's a concern other people will have if that's something that's a topic in this meeting.
And then the next question from Samjin Liu is there's a large duplication of GA1 and A188, and whether or not you found multiple copies of GA1 in some lines.
Yeah, let me click back to our GA1 here. So I think we actually don't have A188 on this chart, which is definitely an oversight. But yeah, we see some weird evidence in some of these where you see like, you can tell here there's kind of these like separate little regions where you see GA1, like it does seem like there's been some sort of duplication here of this region. And also we see some kind of rearrangements. So I had mentioned before this PB860, this new assembly we have, or new genome we have, there's like this tiny inversion in the middle of this, which seems to be part of why there's no female hit in this haplotype. You just see these like male hits with an inversion in the middle that might have maybe just like eliminated the area in which the female gene could be. And we do also see, it's difficult to see on this, or I guess impossible to see on this graph, but we see evidence for some like this Zea luxurians. We've got this female hit here the male hits are so far away that they don't fit on this graph. So we have another version of this haplotype plot, which is like extremely zoomed out and like condenses all these hits into like one tiny corner of the plot. And then the female or the male parts of the hit are all the way out here. And I think that that might be also what we saw in A188. But of course, since I don't have the graph up here, it's you'll just have to take my word on it, which is not great evidence.
So the duplication is about 2 megabase. It's very large. It was not assembled very well in A188, but we know about 2 megabase duplication.
Okay.
I always wonder why. I mean, they need to be 2 large duplication here. Thank you. Very nice.
Yeah, thank you. That's a great point. I will also. It's been a while since I looked in detail at A188. Been trying to focus more on like the molecular evolution side of this recently, and I was out this summer for an internship, so it's always nice to hear details that I've forgotten about. It's a complicated system.
Okay, other questions for Elli? And if not, we have a speaker next week, Regina Fairbanks, and the week after will be Sam Snodgrass. But we currently have no speaker for October 18th. So please either volunteer or strong arm someone in your group to volunteer so that we'll have continued speakers throughout the fall. And let's thank Elli, and we'll see you folks same time next week. Thanks all.
Sweet.